I would like to start actually by thanking for the invitation to be given the opportunity to present my work. So, and what I thought I would present today is I will focus on both analytical work, but also empirical results and look at the molecular signatures of natural selection. And before I look at the results, I'd like to start by providing some background on what kind of different molecular signatures of natural selection are commonly used in evolutionary genomics analysis. And here I would like to focus on the difference between what measures are used within population genomic analysis versus what measures are used within phylogenetic analysis. And if we have two approaches, this means we are actually led to the question, which approach shall we use in which context? Which means which approach shall we use for which study system, but also which research question. And this is a question that has interested me quite a lot over the past years. And together with my collaborators, we have been looking at it first from a mathematical perspective in order to get a better conceptual understanding of what are we actually measuring with the different measures that we see in the genome of natural selection, and what processes influence those measures. And then we have compared our analytical results, or we have not only compared our analytical results with empirical observations, but we also got more information from looking at what do we see in the genome, how can this be modeled. So I think this interaction between comparing empirical observation with analytical results that has been very helpful to get a better understanding of what are we measuring with molecular signatures of natural selection. But let's start with discussing the different molecular signatures of natural selection that are commonly used, and I'll introduce this with our study system, which is the Ficedula flycatcher. And I'll discuss them in more detail later in my talk. Now I'll just use them to introduce the measures. And the measures that I'm introducing are, of course, also applicable to other study systems. But our study system consists of 4 species. And if we now first start with a population genomic approach, that means what is commonly done in population genomic analysis, we have resequencing data. With the resequencing, then that can be for one population or for several populations. And what we commonly do is we compute genetic diversity within the species, which I refer to as pi. Then we plot the variation of diversity along the genome. And then we look for outlier regions. We usually look for regions of locally reduced genetic diversity, which is shown here. You can see my pointer? Yes?
Yes, we can see.
Okay, so which you see here in 3 of the 4 species, you see a locally reduced genetic diversity. Now, this locally reduced genetic diversity is generally interpreted in the sense of a region where there is evidence for natural selection, more specifically a region where there has been a selective sweep. If we find this evidence to indirect, we can also go further and say we want to support or corroborate these results with help of directly measures of selective sweeps, which we also can retrieve from population resequencing data. And then, so here's the same, it's one of the 4 lines, the blue line show the dip in these regions, and then we can see exactly where we saw the dip in genetic diversity, we will find a signature of a selective sweep. So this kind of analysis can be done if we look within one population. If we have the data set that we have, that I'm showing here, where we have pairs of closely related species, we can also go on and look at the comparison of allele frequency differences between the closely related species, which is usually assessed with the measure known as FST. So what we then do, we again plot the variation along the genome, and now we look for regions where we have elevated FST, which happens to be in the same region. So, and this is then again interpreted as indirect evidence for a signature of natural selection. Now I summarize all those measures as within population genomic analysis. Why? Because they rely on population resequencing data. In addition to population genomic analysis, we also have phylogenetic analysis. Now, phylogenetic analysis, we usually apply to distantly related species, so we cannot apply to the data set that I'm showing here but we could apply it to the comparison of the more distant species, and then, so I've taken away 2 of the species, and then we need to add at least 1 outgroup or several outgroups to get lineage-specific estimates in the different remaining species. What we often use to get a measure of natural selection is we apply codon models, and a very frequent measure that I want to discuss in more detail is the measure DNDS, which is a measure of the strength of natural selection. Now, one way to distinguish those measures that I've now introduced is based on the type of analysis, population genomic versus phylogenetic analysis. Another way to distinguish those measures is that with population genomic analysis, we're actually sensitive to signals of linked selection, versus with phylogenetic analysis, we are assessing the strength of direct selection. So that's a second way how we can distinguish those measures. A third way that we can distinguish the measures is if we look at the time aspect. So what do I mean with the time aspect? So if we can start with population genomic analysis, we start with looking at genetic variation within one species or within one population, which is assessed by the measure pi. And every other statistic, like the sweep statistics, are also within species. And then the next level is to compare closely related species, where we look at allele frequency differences between the populations or closely related species like FST, but we can also look at pairwise differences between individuals sampled from 2 different populations like DXY, or we can look at fixed differences between these populations or species, denoted commonly as DS. So here we are now looking at variation between 2 populations or closely related species. Then at the very end of this timeline, we will find phylogenetic analysis. In phylogenetic analysis, we usually estimate sequence divergence. And in order to be able to relate the phylogenetic analysis to the other kind of analysis, we need to actually get an analytical understanding. What is sequence divergence? So and this is what has interested me over the past years. What is sequence divergence? What are we actually measuring? Now, sequence divergence is an approximation of the number of fixed differences between distantly related species. So we're trying to estimate the F. But if we look at it analytically, and I'm going to discuss this in more detail, we are actually, based on the approach we are using, we're actually estimating something like DXY. And then we pretend that DXY is the same as the F. And for this reason, we are focused on distantly related, or we need to focus on distantly related species. Now, these assumptions are actually implicit. The assumption arises from the way how we construct phylogenetic analysis. What do I mean by that? So if we now look at phylogenetic analysis, what is phylogenetic analysis? So in phylogenetic analysis, we compare distantly related species, and we know the phylogenetic relationship among the species. And then what we do is we let each species be represented by a single sequence. That means we ignore polymorphism. So we implicitly make the assumption polymorphism doesn't exist or is negligible. That's an implicit assumption we make by representing each species by a single sequence. And then we focus on variation between species, ignoring variation within species. If we then want to look at this, what does this mean mathematically? This means we are assuming that every mutation that arises fixates instantaneously. This is what's the underlying assumption. Now, how is this assumption motivated? This assumption is actually indeed motivated within the phylogenetic context, because if you look at the accumulation of fixed differences over time, we know that for a fixed difference to happen, we first need a mutation to happen. This mutation will then, if it goes to fixation, it will segregate for a while until it eventually goes to fixation. And this happens on a time scale of the order of n generations. But if we, in a phylogenetic setting, compare distantly related species that are separated over millions of years, then this time scale of n generations is more or less instantaneous, which I tried to illustrate by these stars. So then this means if we have this distantly related species, this assumption makes intuitively sense. because this time until fixation is very short. But if we now are aware of the underlying assumption, we can actually ask the question, which species are distant enough so that we can use classical phylogenetic analysis? This is the same as asking the question, when can we ignore polymorphism within species? Because obviously we don't do it within speciation genomic analysis. And then we can continue with questions like, what happens if we apply classical phylogenetic analysis to closely related species? And can we adjust phylogenetic methodology such that it is applicable to closely related species? And this is the questions that I've been addressing over the past years. And I've been addressing this to start with a population genetic model. And what I'd like to do now is to introduce the basic concepts and ideas of our population genetic model and then turn back towards our questions that I've mentioned. So how does the population genetics model look like? We start from a classical population genetics model where we start from a population of size n. And then we have each individual represented by a sequence, because that's what we do in phylogenetic analysis. Each individual is represented by a sequence. Our sequence is a collection of L independent sites, where the assumption of independence is a classical assumption within phylogenetic analysis. And I'm happy to discuss possible consequences of the assumption after my presentation. So this is how our population looks like. So here I'm illustrating a population of size 8, and then I have a sequence. So each row is a sequence. What we then do, we introduce mutations randomly on monomorphic sites. That means on the first site we could have a mutation happen, but not on the second site, and so on. So for example, now I'm introducing a mutation on the fourth site of the sequence in also the fourth individual. This means, so if mutations only happen on monomorphic sites, we have a max biallelic model. So we don't consider triallelic or more allelic sites. And what we then use, we use the Wright-Fisher model of reproduction with selection in order to model the allele frequency trajectories at polymorphic sites over time. And then, since we're interested in tracking fixations over time, we allow a mutation that has gone to fixation to then mutate further. And this leads to our process, which is a Markov process X, which can be described by 3 terms, J, H, and 1. So what does the J, H mean? So J represents the mutation process. That means, and here on the bottom I show an illustration, that means we are waiting for a mutation to happen in the monomorphic state, and then here a mutation happens. It segregates and then eventually goes to extinction. And then we wait again for a mutation to happen. And then this one happens to segregate and eventually go to fixation. Then the H says, so once it's fixed, we let it go down to 0 because we want it to be able to mutate further. So the H is the path of the polymorphic part. And then the 1 indicates that we put it down to 0 again. So this is how our Markov process looks at one site. Then we have a collection of L sites, meaning we have L of those Markov processes, meaning we have a random field, which I will denote as X and L of T. So what are now the properties of this random field? The properties of this random field are that we know if we let n tend to infinity, we know that the allele frequency trajectories can be described by the Wright-Fisher diffusion process, which allows us to model genetic drift as well as natural selection. We also know that if we not only let n tend to infinity, but we let L and n tend to infinity, our random field converges towards a Poisson random field. So these are properties that we know about our process. What we then do, we have this process, X and L of T. What we do is we keep n fixed, first of all, and we let L tend to infinity, so the number of sites. Then we can show that our random field, we can construct a stochastic integral which we can show for L to infinity tends towards a Poisson stochastic integral. This Poisson stochastic integral is well defined, which means it has finite expected values. It has finite expected values for finite n, but it has also a finite expected value for n towards infinity. And I'm not going to take credit actually for the proof of this, because the proof was derived by my collaborator Ingemar Kai, and the work is published in Theoretical Population Biology. What I now want to discuss is, and why I mention it, I want to use this stochastic integral and the fact that we can have finite expected values. So I want to choose specific functionals f that allow me to get analytical expressions of different molecular measures of natural selection, because that's what we want to investigate. We want to investigate, can we, for example, estimate the NDS for closely related species? So that's our question we have in our text. So we use the properties to get a well-defined stochastic integral, and then we can choose specific functionals f to get derivation of analytical or of our measures of natural selection. So we have population functionals and sample functionals, where population functionals tracks allele frequencies in the entire population would be fixed differences among populations, and sample functionals is differences between samples, which is what we actually do in a phylogenetic setting. We have a sample size of 1. So we then choose specific functionals, and then we get fixed differences between populations or between samples. If we have a sample size of 1, that's actually what we need for divergence, because in a phylogenetic setting, we have a sample size of 1. So that means we now get an expression of sequence divergence D as a function of time, which, as expected, has a linear contribution. The more time passes, the more fixations accumulate over time, which is what we expect in a phylogenetic setting. What we now also show is that we have a nonlinear contribution due to polymorphisms to sequence divergence. What we then can show is that if we let t tend to infinity, then the contribution of polymorphisms to sequence divergence becomes negligible. So that simply means that indeed the assumption that we make within phylogenetic methodology that we ignore polymorphisms is well justified for distantly related species, which is also what we discussed intuitively. It makes sense. But now with our analytical definition at hand, we can address the other more specific questions. What is the time scale that this assumption is justified? And what happens if we apply phylogenetic methodology to closely related species? And this is a question we have addressed now, not for sequence divergence, but for our measure of natural selection in a phylogenetic setting, that is the NDS. And the NDS is sequence divergence for a category of selected sites, that's the non-synonymous sites, divided by a category of neutral sites or neutrally evolving sites, that's the synonymous sites. So we estimate, so the NDS, I call it an estimate of omega, where omega is our target parameter. I use omega because this is how it is called in many of the phylogenetic methodology that is available. So I will use the terminology omega as a target parameter of what we want to measure, our fixation rate ratio. TNDS is the sequence divergence of non-synonymous over synonymous sites, which we derive from our phylogenetic methodology. And we use this now, we derive an expression of the NDS based on our model. For the selected category, we introduce variation in selection across sites using a distribution of fitness effects, which is a common way to describe variation of selection among sites. So we do that, and then here we see the analytical results of this. So here on the black dashed line, you see our target parameter omega. This is what we want to measure. What we actually measure, and now here on the x-axis you see time scaled in n generations. So and I would put a guess, human chimpanzee at around 5. Well, that's of course not precise, but maybe you can have an idea what this means. what we see here in the red line is we see that the estimate, the NDS, is actually hugely biased for short divergence time, and it's still biased quite a lot for divergence of human and chimpanzee, and it varies off quite slowly, but it converges towards our estimate. What we then wanted to know, so this is simply what I just said, what we then wanted to know is if we sample several individuals, and if we then exclude all the polymorphic sites, can this improve our estimate of omega? So we get expressions where we now not only sample one individual, but we sample several individuals. So we have also some dependence on sample size. And if we do that, and now I'm increasing sample size, so from 1 is still the same red line, 2 would be 1 diploid individual, and so on. So we have until m equal to 8, and then we use m to infinity, which converges towards sampling the entire population. So what you can see here is that indeed our estimates get better and better. But what is also important to realize is that it doesn't become unbiased. We still have a biased estimate for omega for short divergence time. And why is this the case? Because in the very beginning, when species diverge, we are not actually measuring lineage-specific fixations. What we are measuring is lineage sorting. So we are measuring the rate of non-synonymous over synonymous lineage sorting and not fixations, lineage-specific fixations, which is the reason why sampling more and more individuals will not help us if we simply exclude polymorphic sites. What we need to do is, if we want to get a correct estimate, is we need to model polymorphism within phylogeny. This is the only way how we can correct it, or we cannot correct it by simple exclusion. So to conclude this first part is that we can conclude that current phylogenetic approaches for the estimation of omega are only applicable to distantly related species, something we already understood intuitively. We can also show that exclusion of polymorphism really improves the estimate of omega, but it doesn't lead to an unbiased estimate. What we now need to do is, and what we are working on, is we use our mathematical framework to develop codon models at the interface between population genetics and phylogenetics. So we modeled polymorphism within phylogeny, and I will get back to this part at the very end of my presentation. What I now want to focus on, I want to emphasize that the mathematical framework that we are using is actually a very flexible mathematical framework, so we can incorporate more population level processes to study their impact on molecular measures of natural selection. And that's what we are doing next. So in order to do that, we need to first of all look at the model assumptions that are underlying our mathematical model. So we assume constant population size, we assume no migration, free recombination, we have a fixed distribution of selection effects, and we ignore at the moment molecular processes such as multinucleotide substitutions and GC-biased gene conversion. We mentioned that GC-biased gene conversion is a process that has interested me a lot over the past, and we're working on extending most of the assumptions, but what we have already results on, and what I'm going to focus on right now is to relax the assumptions on constant population size. So if we do that, what does this mean? This means our target parameter omega, which has been constant over time, will now no longer be constant through time. It will change with the change in population size. And what we want to know, how does it change? And this is the PhD thesis work of a PhD student, Rebecca Müller. And what she has been working on, she has been introducing a change in population size over a time interval. So we are considering omega over a time interval 0 to t. And then some work
Can someone hear something? It got lost for me.
It's same. Carina, are you there?
Yes.
You're off for a bit.
You hear me again?
Now?
Yeah, that is working.
So where did it stop?
I guess when you were just starting to explain this slide.
Okay, so
just introduce Rebecca, and then we lose you.
Okay. Then I will study. So what the PhD thesis work is about is that we study a step change in population size in the time interval 0 to t. So we consider a time interval 0 to t, which could, for example, since we're interested in phylogeny, be the branch length of a lineage-specific branch in a phylogeny. So we have a time interval 0 to t. Within that time interval, we introduce a step change in population size at a time t star. This can be at the very beginning, it can be at the very end. It's more or less our choice. We can put it where we wish to put it, and then we consider decrease in population size for kappa smaller than 1 or increase in population size for kappa larger than 1. So then we have this step change for the different kappas. That means we can now describe our right-Fisher diffusion process by introducing our parameter kappa into our right-Fisher diffusion process after the time of the step change, or from the time of the step change. And this is what we have been doing. So how does our allele, or do our allele frequency trajectories now look like? So we have 3 different passes. So we have entirely blue passes that are passes that are going to fix. So, and then I only highlight the fixation since we're interested in fixations over time. So the blue passes are going to fixation before the step change. Then we have passes that are entirely red, mutations originating after the change and going to fixation after the change. And then we have blue and red passes, those that happen before the change and are segregating after the change and going to fixation then. So this means if we then derive the fixations over time, we will have 3 contributions. We will have a contribution from the blue passes, the red passes, and the correction term for those passes that are blue and red. Now, this relative contribution of the blue and the red, of course, will depend on where in the time interval we put our change. So if it's in the very beginning, we will have a smaller contribution of the entirely blue passes and a larger contribution of the red passes. This also highlights that what we're considering is an accumulative measure. So everything that happens in the time interval is of relevance. And if we have a more recent change in population size, then what will matter is mainly what happened in the past before the change in population size. I'm going to emphasize this because I think this is important. Why? Because if we now look at our target parameter omega, which is what we are modeling over time. So here now I place a change in population size at t star equal 1. So our parameter omega for a decrease in population size, which is highlighted by these pink colors and an increase by the green colors, you can see, and that's now what you need to look at is these dots, like the triangles. So that's our derivation. What I'm showing here by the dashed lines is if we would ignore the contribution of polymorphisms, and now I'm turning back, if we ignore the contribution of polymorphism, this term is negligible, then we only have the weighted mean on the time interval. And this is the dashed lines. But what we can see here is if we ignore polymorphisms, it still takes a while until the estimates converge towards the new equilibrium. But if we actually don't ignore polymorphism, then it takes even much longer for the estimate to converge. And overall, we can see even though the blue interval is really short, so we have 20 time steps and the blue interval is only 1 time step, we see that it takes quite a while for the estimate to equilibrate to the new equilibrium. If we then, for example, would place our change at t star 18, so we have 18 time steps of the blue interval, then it becomes actually quite pronounced. Then we almost don't see any signature of the change in population size in our estimate of omega. And this is important for data analysis, for example, because what we're often interested in is what happens if a population moves to an island. It has a smaller population size, but if this happened very recent, we will not see it in our estimate of selection. So then we cannot actually, if we use omega as an estimate of selection, we will not observe a change in the efficacy of selection. We only observe changes in the efficacy of selection if we use omega, if the change in populations has happened quite in the past. So we thought that omega is maybe an unsatisfactory measure to track recent changes in population size. So then we look back, what can we use if we don't use omega? We can rely on nucleotide diversity pi. What you will, and I illustrated in or highlight it in orange. Why? Because I have here these orange passes to illustrate that's currently segregating polymorphism that will matter. It doesn't matter what happened in the very past, only the currently segregating polymorphisms. And then we cannot have, of course, passes that cross the line of T star, depending if the mutation originated before or after the change in population size. But that means what matters is the pi after the change in population size plus a correction term for those passes that cross T star. And what we then do, since we want to measure selection, like for D and DS, we now compute pi and pi s, which is the ratio of non-synonymous over synonymous diversity. And if we do that, and we plot now, so here we have our omega on the left and our pi and pi s on the right, you can see it converges much faster. Then, if we now look at if we plot or have the change in population size at time T star 18, then we saw that omega was very insensitive. For pi and pi s, since it's not an accumulative measure, we can simply put the line 2 time points or 2 time steps after the change in population size. So we would be here. So we can see even for recent changes in population size, pi and pi s gives us a much more reliable information on the efficacy of selection after the change in population size than omega would do. So this now tells us that, well, I like to highlight that the Poisson-Random field model actually are very useful and allows us to describe the allele frequency trajectories after change in population size, or by incorporating also we can incorporate also other population level processes. So we have then formulated a non-equilibrium allele frequency spectrum during non-equilibrium periods, which then has allowed us to derive macro as well as micro evolutionary measures of selection, where macro would be our phylogenetic measure, the NDS, and micro would be our measure pi and pi s. This then shows that the macro evolutionary measures react really slowly to change in population size. And if we want to capture changes in the selection efficacy after recent demographic changes, we do better by looking at pi and pi s. Then we can, of course, also use our mathematical model to incorporate other population level processes also among divergent species, because now I've only discussed it within one species, and this is currently ongoing work that Rebecca is working on. So we are now also modeling change in population size at the onset of speciation, because we now want to model this in a phylogenetic setting. So we need to consider at least 2 species. We then also model migration between the pair of diverging species, and we are looking at different ways of modeling variation in recombination rate across the genome. But this is ongoing work, so I'm not going to discuss any more details about this. I would now like to use the final part of my seminar to look at what we actually observe in our data. And to do that, I'd like to look at our study system, the Ficedula flycatchers, and discuss some of our observations in the Ficedula flycatcher system. So what are the Ficedula flycatchers? The Ficedula flycatchers are birds, passerine birds. And the Ficedula flycatchers are actually a model system of speciation research. Birds are generally popular within speciation research because they're easy to observe in the field, so one can collect behavioral factors as well as ecological factors that are relevant to speciation. And this many of my collaborators or ecology collaborators at Uppsala University have been collecting for several decades. There's numerous of behavioral and ecological information available, which makes them an interesting study system. And there is also genomic resources available for these 2 species that I'm highlighting, the collared and the pied flycatcher. We have a reference genome for the collared flycatcher, and we have resequencing data also for the pied flycatcher. What else do we know about the collared and the pied flycatcher? We know that the collared and the pied flycatcher are part of a species complex of 4 black and white flycatchers. Their closest relatives are the red-breasted and the tiger flycatcher. So what we next did with a PhD student, Madeleine Chase, what we did is, in addition to the samples that we had available for the collared and the pied flycatcher, we now added samples for the red-breasted and the tiger flycatcher. And we chose those species because we wanted to study 2 independent pairs of closely related species so that we can assess measures of natural selection at different time scales. We can look within population, between closely related species, and we can use a phylogenetic setting. Another reason why we wanted to have 2 independent pairs of closely related species is because we wanted to understand the differentiation landscape within a speciation genomic context better and see how they are evolving in 2 independent pairs, independent in the sense of that the phylogeny is well resolved and we don't have any signatures of incomplete lineage sorting or ongoing gene flow between these pairs. So from the red-breasted tiger pair to the collared and pied pair, there is no gene flow among those. So we have these 2 independent pairs of flycatchers, and we want to perform, first of all, population genomic studies to start with, and then we want to continue with phylogenetic analysis. So what we did first is we wanted to see, are those pairs indeed independent? So we wanted to look at the number of shared polymorphisms, and we saw that the 2 pairs share only 2% of polymorphisms, so they're really independent. And as I said, we also don't see any signatures of incomplete lineage sorting or gene flow among the pairs. What we then did is we computed different population genomics statistics, pi, selective sweep statistics, FST, and DXY along the entire genome. And of course, you cannot see very much in this graph, so I've chosen 2 different chromosomes, chromosome 8 and 11, because I think it's efficient to make my find. What I want to highlight is what we saw in this system is that we saw that several of the FST peaks that we see in one species pair are also found in the other species pair. And this is what is kind of rather particular or interesting to birds, is that they show many shared signatures of FST peaks, which is shown here on chromosome 8. But we also found several lineage-specific signatures, which, for example, is highlighted here on chromosome 11. Now, this means if we have this observation, we can ask the question, what explains the presence of shared versus lineage-specific FST peaks? Is there anything that can explain why some are shared and some are lineage-specific? So to do that, we first of all used also the other measures of assessing molecular signatures of natural selection. So we looked within species and looked where we see a region of elevated differentiation. We generally find in at least one species of the pairs a region of reduced diversity. So here you see reduced diversity in 3 of the 4 species, which leads to a peak in both pairs. In this region, and now a bit, we see only a peak in the collared pied comparison, and we see a reduced diversity in the collared, so the blue line. So that means what we can see, and these peaks, these dips in diversity are generally a result of a selective sweep. So I also want to show that this is also the case. So we see that where we see this dip, we see a selective sweep for both chromosomes. And this is then valid now for all the chromosomes, but I'm making, and then this was then in the next species, the pied flycatcher, we also see, if we see a dip here, we see a peak here. Here we don't see a dip, we don't see a sweep, but that's not valid for all the species. So what we can conclude is that we find signatures of selective sweeps in both in shared as well as lineage-specific FST peaks. Now this tells us selective sweeps are not the driving force for lineage-specific peaks. And why did we address this question? Because this is a common hypothesis. A common hypothesis is that shared peaks are a result of background selection. So we have reduced diversity simply because of strong background selection. And lineage-specific peaks are then a result of lineage-specific adaptation and positive selection. What we show here is that no, this is not the case. We actually find signatures of selective sweeps in those kind of peaks. So we say if not lineage-specific adaptation and positive selection. What is the driving force of the lineage-specific peaks? And in order to do that, we need to remember that FST peaks and reduced diversity, we are assessing signatures of linked selection. So we see a strong dependence on recombination rate. So what we then did is we estimated recombination rate in 2 of the species, 1 in each pair, and then we see a clear relationship with FST in that pair. So we have a recombination rate for the taiga flycatcher available, and then we see a clear relationship to FST red-breasted versus taiga, and the same for collared versus pied FST. We see a clear relationship with collared recombination rate. So we said maybe if the reason why we see lineage-specific peaks is because we have lineage-specific changes in recombination rate. And to investigate this, we then plotted the estimate of recombination rate for the different peaks. So we have a background level, no peak, and then we have shared peaks, peaks unique in the red-breasted taiga, RT, or collared pied, CP. And what we see is that if we have a shared peak, then recombination rate is reduced in the taiga. It is reduced for the RT unique peaks, red-breasted taiga, but it's not reduced for the CP unique peaks. The recombination rate that is reduced for the CP unique peaks is the collared recombination rate, which is shown here, but not in the RT unique peaks. So this really tells us that we find evidence, and it's shown now here in the last line, we find evidence that lineage-specific FST peaks are driven by evolutionary changes in the recombination landscape. Not, so at least we don't find evidence in our flycatcher system that lineage-specific FST peaks are not the result of lineage-specific adaptation, because sweep signatures are found also in the shared peaks. So what we then wanted to do, we wanted to complement these results by also performing phylogenetic analysis, so that we can see where we see signatures of linked selection and signatures of selection based on population genomic analysis. Do we also see signatures or increase the evidence for adaptive evolution in those regions? So we computed DNDS, but we also wanted to combine it with PI&PS, because we know from the analytical work that DNDS is not very sensitive to demography. So we also wanted to complement it with PI&PS. So our question that we wanted to address is, do shared peaks show higher rates of adaptive evolution? Why? Because this is a region where we see sweeps in both species. So it seems to be recurrent sweeps that are found in the shared peaks. So we wanted to know, do we also find higher rates of adaptive evolution in those peaks? The shared peaks as compared to the lineage-specific peaks, but also as compared to the background. So that's what we did. So we looked outside the peak region, that's our background. We looked in the shared peaks, and we looked at the lineage-specific peaks. We computed PI&PS and DNDS. And by combination of these estimates, you can also compute the rate of adaptive evolution omega. What we see now, if we only look at the taiga flycatcher, we would see, yes, indeed, in the shared peaks, we find elevated rate of adaptive evolution. But, and that's very nice that we have 2 species now, because this is like a biological replicate. We can see we don't find evidence in the collared flycatcher. So if you only find it in one species, it's maybe not something that should be interpreted in terms of elevated rate of adaptive evolution in shared peaks. So we don't find any consistent evidence that there is higher rate of adaptive evolution. In fact, and what we see is that we can interpret our signatures completely based on differences in demography between the 2 species. Because we know that our estimates, PI&PS and DNDS, are sensitive to demography, and in particular, the estimate that is based on the combination of both omega A is highly sensitive to the demography. So we wanted to know more. What about demography? Does population demography influence our molecular signatures of natural selection in the flycatchers? And this is now ongoing work. But what we also know is that, for example, we know from the collared flycatcher, it has recently populated the Swedish island of Gotland. At least that's where we have collected our samples. So we know that, for example, recent demography not only influences PINPEST, but DNDS, but very recent demography is actually strongly influencing signatures of linked selection. And we see that where we see a very recent bottleneck. So now I'm listing the number of selective sweep signatures that we find in the 4 populations. And we can see that we have many fewer selective sweeps after a recent bottleneck. This is simply a matter of power, or LD breaks down. But then we are also complementing this work. So we are estimating demography also over larger evolutionary timescales with classical models like PSMC, but we are also using demographic modeling. And this is now the work of Madeleine together with a visiting student who visited our lab. And as I said, this is ongoing work, so I don't have many results to show, but we can actually interpret our results that we find. They make sense if we interpret them together with demographic history. With that, I'd like to thank in particular Ingemar, Rebecca, and Madeleine, who have actually conducted most of the analysis that I've presented today. I'd also like to thank Hans Ellegren, who was my PhD and postdoc advisor, and then other collaborators, Maureen, our visiting student, and then Jochen, Verena, and Fidel, who also let us test our theory in their data set. That's the CROW data set. I'd like to thank funding. And before I would end, I'd also like to take the opportunity to actually announce or advertise a postdoc position. So as I said, I still have used the logo from Uppsala University in my presentation, but I just this month moved to the University of Lyon, where I'm now working at the Laboratory of Biometry and Evolutionary Biology, LBBE, and we are now together with Laurent Guéguin, who is a computational scientist working at the LBBE. We're now looking for a postdoc to join our work, where we are currently developing software so that we can model polymorphism within phylogeny in order to get unbiased estimates of selection based on the combination of intra and intergenomic data. So we have funding for 2 years available if you find this interesting, and if you are interested in software development, statistical inference, and mathematical work. We are happy that if you contact us, so you can contact either me or Laurent by email. And the project also involves then some more data analysis. And with this, I'd like to thank again for the opportunity to present my work, for the opportunity to announce this postdoc position, and thank you for your attention.